Namaste. Welcome to Sharps Biotech for you. Today we'll discuss about polymerase chain reaction. It is developed in 1983 by Carrie Mullis, an American biochemist. It is a technique in molecular biology that produces several copies of certain DNA fragment. The principle behind PCR. It is based on enzymatic replication of DNA, where DNA polymerase synthesizes new strands of DNA complementary to the template DNA. The nucleotides are added to the 3' OH group only. Coming to the components. The first, DNA template. It is nothing but the DNA of interest from the sample. Then we have Taq polymerase. It is a DNA polymerase that is isolated from Thermus aquaticus that lives in hot springs and is active at 70 degrees Celsius. It is thermostable and does not denature at very high temperatures. Then we have oligonucleotide primers. These are the short stretches of single-stranded DNA that are complementary to the 3' ends of both sense and antisense strands of nucleic acids. Then, deoxyribonucleotide triphosphate. These provide energy for the polymerization and are the building blocks for the DNA synthesis. These are single units of bases. Then we have buffer system. Magnesium and potassium provide optimum conditions for the DNA denaturation and renaturation. It is also important for the fidelity, polymerase activity, and the stability. Fidelity is nothing but the ability to accurately replicate. Then we have types of PCR. The first one is real-time PCR. DNA amplification is detected in real time using fluorescent reporter. Signal of fluorescent reporter is directly proportional to the number of amplified DNA molecules. Then we have nested PCR. It is designed to improve sensitivity and specificity. There is reduction in the non-specific binding of products due to amplification of unexpected primer binding sites. Then we have multiplex PCR. It is used for the amplification of multiple targets in a single PCR experiment. Then we have quantitative PCR. It uses the DNA amplification to detect, characterize, and quantify a known sequence in a sample. Then last is arbitrary PCR, arbitrary primed PCR. It uses primers, the DNA sequence of which is chosen arbitrarily. Coming to the steps of PCR. Here is the double-stranded DNA, which is subjected to denaturation at 96 degrees Celsius. The reaction is heated strongly to separate or denature the DNA strands. This provides two separated single strands, which act as template for the next step. Then we have second step, that is primer annealing, which takes place at 55 degrees Celsius. The reaction is cooled so that the primers or the short stretches of single stranded DNA can get attached to their complementary sequences on the single stranded template DNA. Then we have primer extension that takes place at 72 degrees Celsius, where nucleotides get added in this sequence, in this direction on the complementary strands of DNA. The reaction temperature is raised so that Taq polymerase extends the primers, synthesizes new strands of DNA. So, this is the result of first cycle, where we have two DNA molecules. Hence, the number of DNA molecules has doubled. This takes place for about 25 to 35 times in PCR, which takes about 2 to 4 hours. Now coming to the applications of PCR. First is in medicine. Testing of mutations, monitoring the genes in gene therapy, detecting disease causing genes in the patients. These are the applications under medicine. Then moving on to forensics. It is a tool in genetic fingerprinting. It helps in identifying criminals and also during the paternity tests. Then coming to the research and genetics. to compare genome of two organisms in genomic studies, phylogenetic analysis of DNA from any source such as fossils, gene mapping, and analysis of gene expression. Then moving on to the advantages. PCR is so sensitive that DNA present in an individual cell can also be isolated and amplified. It is a faster technique and it is less tedious. With this, I wind up today's session about polymerase chain reaction. Please like, share, and subscribe to my channel.